Bit like Scott, this was hard to fit in all the cool stuff that we do into the amount of time. But luckily, Shuang actually gave you a great highlight on her work, so I may be able to skip a couple of the slides because you've already seen that. I wrote a slightly ambitious title, and I don't think we're going to get to the climate change part, but what we are going to talk about is our understanding of climate drivers in water systems broadly, because to understand climate change, we need to understand the climate drivers in the current, historic climate. Let's go. Done that. As water managers, as humans in the world, it's the extremes in the hydrologic cycle that really keep us awake at night. And so that's the floods, it's the droughts, and then it's the water quality impacts too. So here in algal bloom, although there's a whole range of different, ecological impacts from water quality issues. And so these are not our everyday, we hope, regular occurrences. They occur by definition infrequently. And so therefore, this talk is really about kind of complete opposite of Scott's problem. I wish I had hundreds of thousands of data points to fit distributions. We have very few. And that's partly because our instrumental data is very sparse in space, but also very short in time. So, daily rainfall data, we've got about 8,000 gauges across Australia where we've got more than 30 years of data, but we can see massive gaps in our record. If I come down to sub-daily rainfall data where, gives us the majority of our flood risk in urban areas, we're down to about 2,500 gauges that have 8 years of data. Streamflow data is managed by a whole range of different organizations around Australia, which gives us lots of challenges in terms of pulling it even into one consistent, coherent database and, varying lengths as well. So that's the pure hydrology side of things. But if I come into other parts of that affect the hydrologic cycle, you can see down in the bottom left, the carbon flux data from eddy covariance stations run by Turn. We've just got a handful of those across Australia. And it's very hard to then say something about other catchments. And then water quality data, this map is from the Bureau of Meteorology, which does bring in some water quality data from around Australia, but not in a consistent way. And so this is pH data, but you can see that in space, it's very sparse. We can't say a lot about what's happening in other locations. And then because we're interested in extremes, I've only got 8 years of data in my rainfall data, or the same with water quality. It's very hard to estimate those extremes with reasonable bounds of uncertainty. So what do we do? So we really, as in this kind of space, we're really going to be look at 3 different alternatives. And so I'm going to give a highlight of that. Now you can tell that I'm part of DARE as a domain expert, not a data scientist, because there is not a single equation in this presentation, but Carolyn even put one in hers. So I feel like I have really let the team down here. So we're going to talk about some different alternative data streams, the methods that might allow us to best leverage this sparse data that we've already got, and then modeling that might help us understand our processes better. But at the Melbourne water level, Shuang did have some success in explaining the sorts of trends that we saw in the colour here. And so you can see these sort of climate affected water bodies, and then ones where climate was not such a strong driver. And so although here we don't have any coherent spatial patterns, it's not hugely surprising because these water bodies all have very different catchments driving them. They're different sizes. And so their susceptibility to climate is going to be different. But what we found is that when they were climate affected, temperature and rainfall over the last month drove water color. A measure of drought over the last kind of year was important, and then a little bit around temperature and rainfall extremes too. Interestingly, climate drivers in this context, so things like the El Nino Southern Oscillation, the IOD, the things that Delaney was talking about, were not important drivers of these systems, and that's probably because they're really small. And so the size of our water bodies tells us a lot about the temporal periods of its memory. And so in this particular context, this is not so, not so influential. So with our, with this, the reason we went into satellite remote sensing was, as Shuang explained, was we had very sparse records and very sporadic data collection for the sites where we did have records. And again, it was only, sort of 5, 10, maybe 20 years of data. And now that's not really enough to understand long-term climate variability and to put the historical period into even a longer-term context. So we've been doing some really cool work looking at the potential for tree rings to help us understand very long-term climate variability. So this is my PhD salmon in Canada in the middle of summer being attacked by mosquitoes, hence all the netting, but learning how to collect tree rings. And so what we do in this work is try to pick, so the tree rings capture variations from year to year in wetness and temperature, because the trees grow better under good conditions and they grow slower, and therefore the tree rings are thinner under worse conditions. Turns out there's not that many trees in Australia that collect this information because things aren't very cold here, and it's, you know, the trees are kind of evolved. And so Australian tree rings aren't great predictors of climate. There's a few that are, but what we can find is places around the world that have the same drivers, those ENSO and IOD that we were talking about before, that pick up a signal from those, and then we can use those remote teleconnections to say something about what's happening for Australian catchments. So my postdoc, Philippa Higgins, has worked with a great team of researchers during her PhD to see how we can use tree rings, remote tree rings, to better understand Australian hydroclimate variability. And so her recently published paper, we reconstructed 800 years of stream flow for the Murray-Darling Basin using remote tree rings. So she used all these tree rings shown in these red dots around the Pacific Ocean, so places that are also affected by ENSO, which is the major driver, as we just saw, of variability in the Murray-Darling Basin. And what she was able to do was she took, so this is our instrumental period here in the black period, and she was able to use a nested principal components model, regression model, to take the tree rings and the stream flow during the instrumental period, and then the tree rings go back 800 years, and so she was able to use that model to reconstruct the past stream flow. The really cool thing about what she did was then she took climate model projections through to the future, which is shown at the right-hand end in grey, and contrast the droughts that we got from that to ones that we've seen in the last 800 years. And so what she sees is here, the black dot in both of these plots is the millennium drought. And what we've got here are two different ways of measuring drought. Well, there's three different measures of drought severity. So one is the magnitude, how deep the drought is. One is its duration, which is the x-axis in both these graphs. And one is the peak. Sorry, the magnitude is the cumulative sum of the drought deficits, and then the peak is how bad the actual peak is. So what we can see is that historical droughts, even in the 800 years, the millennium drought is one of the worst in the 800 years of record. The black dot is far to the right, which is bad from a drought perspective in 800 years of data. But with climate change, we will see worse droughts from these climate models. And so this is based on the Bureau's Australian Water Outlook data. So here she fitted probability distributions to these sort of bivariate problems to look at these different aspects of drought together. The next thing I wanted to talk about was how we might make best use of the sparse data that we've got. And so this is work by our PhD student, Mariam, who's been working with Water New South Wales through her DARE PhD, and with Lucy Marshall and myself. And so what the first part of her PhD was looking at increased periods of algal activity in Lake Burragram or Warragamba Dam, a major water supply for Sydney. And Water New South Wales regularly measures chlorophyll A, which is a measure of greenness of the water. Now, chlorophyll A doesn't mean necessarily that there's an algal bloom that is going to be problematic, but when it is above a threshold of 5 milligrams per litre, then extra lab analysis is done to work out what species of algae are available and what that means in terms of supplying water to Sydney Water. Are there going to be problems further downstream in the treatment train? So it's very important for Water New South Wales to kind of know about these higher levels of chlorophyll A. But you can see from our time series that although we've got sort of 20 years of data, there's actually very few time steps where we've had high chlorophyll A. So what Mariam looked at was a class of problems, a class of solutions, sorry, to this problem called resampling techniques. And now Rohit, one of the CIs from the domain science side, spends a lot of his time thinking about resampling methods. And so we sort of took some of these ones that are already in the literature and tested how they worked out in this problem. So we had 3 SMOTE where we're essentially, so in resampling methods, we're trying to solve this problem that I have fewer points in the gray side than on the white side of the line. So in the SMOTE side, I create extra synthetic data, or Mariam does, I don't do it, so that there's more points in the gray bit than and about the same as the number of points in the white bit. Or I can take points out of the white bit. So a bit like, in the previous presentation, like we could just leave out the missing data, but here we could just leave out some of those points in the white area, or I can use a combination of the 2. So Mariam tried all 3 of these methods. And what we've got here is the skill of the model. So what we want is a good true positive rate and a bad false positive rate. We want to correctly identify high chlorophyll A's, and we don't want to misclassify data. So when we took our original imbalanced data set, basically her Bayesian net could never find the high chlorophyll A's. It had a 0 true positive rate. It was a shocking model. But when we used these resampling techniques, then we could find them. But the trade-off is that we also then have false positives. Now, in this particular context, maybe that's not such a bad trade-off. Like it's better, it's more conservative to think we might have high chlorophyll A than, to miss it completely. And so these are kind of the trade-offs we can get by creating extra data samples in the gray area. The other thing, though, that we got from this, so we used a Bayesian network. And so there's sort of 2 steps of this. One is to construct the Bayesian network, taking a whole lot of predictors and trying to predict chlorophyll A. So we had our original data that had no success in predicting chlorophyll A. Unsurprisingly, all of these predictors that we know from talking to Water New South Wales and from our own knowledge that should affect chlorophyll A, the model couldn't find any of those relationships. And the only relationship it said was chlorophyll A impacts dissolved oxygen. When we take the rebalanced data, though, what we find is a much more complex structure to our Bayesian network that's starting to look a little more reasonable, that temperature should affect chlorophyll A. We know that algae is more prevalent in warmer temperatures, that nutrients, nitrogen might affect it, dissolved oxygen and conductivity are now affecting chlorophyll A. And so by resampling, we were actually able to find more true physical relationships from our resampled data. Which kind of brings me to our last point, which is how do we then use models to improve our understanding? So my last example that I wanted to talk about is some work by Lucy and my amazing PhD student who's now a postdoc at Western Sydney University, Claire Stevens, who's done a whole lot of work around catchment modelling. But normally our catchment models are really simple. We kind of have rainfall in a bucket and then streamflow comes out. But they give us surprisingly good predictive skill. But we don't know why. Like there's nothing in there that actually reflects true processes. And if we're going to model under climate change and we think that ET might change, well, that's going to be a bit of a problem. So Claire set up a detailed eco-hydrologic model called RESUS for this catch. She's done it for a few catchments, but this is an Australian study we've just published for a catchment down in Victoria, the Upper Yarra Reservoir. And so the work that Monica presented before, I was like, oh, this could be really interesting on this changing kind of states, because what we've seen in these catchments in Victoria is during the Millennium Drought, streamflow went down. This is not surprising. There was less rainfall. But what is surprising is the ratio of the amount of rainfall to the amount of streamflow also went down. So that's the bottom plot here. So you can see that prior to the Millennium Drought, we tracked along at sort of somewhere between 20 and 40% of rainfall became streamflow. But during the Millennium Drought, we never got those big ones. It was all down below 20%. And what's really interesting in some of these Victorian catchments is even post-Millennium Drought, the catchments have not recovered their streamflow ratio. And this is a big problem for water managers because this conversion of rainfall to streamflow is vital for filling dams. So what Claire did was able to see that when she used this detailed ecohydrologic model, was here we've got three things. So we've got the stream flow, which is what we normally model and measure. Then we've got evaporation, and then we've got the transpiration, what the trees are doing. And so what she was able to show was that the big decreases in stream flow were because although the rainfall went down, the trees didn't change the way that they were breathing. And so they just kept breathing lots. And so then the runoff went down further. And so this is really interesting because people had previously explained a lot of this as around groundwater interactions. So this modeling-based study shows that, you know, vegetation is going to be really important for us to understand runoff transitions in the future under climate change. And this just shows that the detail of this model, where we model spatially as well, these are maps of the catchment. So you can see that the actual evapotranspiration in the streams is always higher than on the slopes. But during the drought, it actually increased in the streams at the you know, although the slopes decreased. And so these are sorts of models that we would never normally look at in hydrology. And so they're very computationally challenging. We need a lot of data to run them, but they do help us understand the processes a lot more. So there are still lots of processes that we don't understand, and that data sparsity in time and space and our process understanding is still challenges, but that hopefully a mix of modeling and data analytics will help us answer these questions going forward.